Thank you. So, well, as Anna already said, today I'm going to talk about fungal competition and partner choice in the ectomycorrhizal symbiosis. And this is part of the work that I did at the Hawkesbury Institute for the Environment in the Western Sydney University. So, Anna already said this, but for me it's very special to be here because this is where everything began for me. So I did my undergrad studies at this university, then my first internship at CREAF with Anselm, and I also worked for 2 years at the botany unit. So, but this was many years ago, as Anna said. And then, So then I did my PhD at IRTA with Xavier here, and a first postdoc at INRA in France, another at CSIC in Almeria, and then I moved to Western Sydney University. And after almost 5 years there, we decided to come back to Barcelona, so here we are. And I also transitioned a bit towards more of a management career. So now I work for the government through the CSUC, the Consorci de Serveis Universitaris de Catalunya. So it's 1 year that I quit academia, so be kind with me. And so all this to say that it's been a while since I was here, but it still feels a bit like home. So thanks for having me and for the invitation. And yeah, of course, thanks for the organizers to make this possible. So, well, as I said, I spent the last 5 years working as a research fellow of the Hawkesbury Institute for the Environment. So some of the people already from CREAF already know this place, some have even been there. And I can only say it's a great place with great people and excellent facilities to assess global change impacts on ecosystems. And I was very lucky to be involved in several of these big experiments, such as the Uke phase here or the whole 3 chambers experiments. And particularly, I was interested on what's happening in the soil. So in Australia, that's lots of fun and also quite an experience, given all the animals that can kill you while doing your field's work. But it was quite fun. And so I'm particularly interested on the mechanisms that drive the soil microbial community assembly, and then also on plant-soil interaction. So the influence of soil microbes on plant community dynamics, plant performance, or plant response to environmental change. And among soil microbes, my main focus has been ectomycorrhizal symbiosis since my PhD. And these ectomycorrhizal fungi colonize the roots of most tree species, forming beautiful mycorrhizas, such as these ones here in eucalyptus roots, that form a thick mantle around the root cells. And these fungi get carbon from the host plant, and in exchange they provide several benefits, such as access to nutrients and water through the extensive mycelium that they form in the soil to gain access to nutrients, and then also protection from soil pathogens or tolerance to heavy metals. So these fungi play a key role in forest ecosystems, and so understanding their response to global change and integrating it into predictive models is highly important. For example, Terre et al. Show that mycorrhizal types of whether a plant associates with arbuscular mycorrhizal fungi or ectomycorrhizal fungi, it was one of the main predictors of the CO2 fertilization effect on plant biomass. But it's also quite challenging because the 2 components of the symbiosis, the fungus and the plant, can respond differently to the modified factors. And moreover, they will have a direct response to the factor, but they will also have an indirect response of what's happening on the other partner. So, and in addition, it has been shown that different ectomycorrhizal fungi respond differently to climate factors. So some individuals will perform better under the new conditions, while others will perform worse. And this is interesting because multiple ectomycorrhizal fungi compete to colonize the root system of a plant. For example, here you see several examples of ectomycorrhizal fungi that coexist on eucalyptus roots. So if some of them respond positively to a modified factor and others respond negatively, the balance of their interaction might shift, which may have consequences on community structure or functioning. And last, the expected increase in plant growth under new scenarios will result in higher nitrogen and phosphorus demand by the plant, so higher nutrient limitation and higher competitive pressure between ectomycorrhizal fungi. So all this is why I'm interested on assessing the impacts of climate change on the competitive interactions. For example, Sara Hortal in saprotrophs show that warming affects the competitive outcomes. Here, each color represents one species, and you can see that with a 3 degrees increase here in red, there is a change of the dominant species. Also, while analyzing community composition, we have also seen changes in the patterns of the community under different factors. I don't know if this is gonna work. Yeah. So with elevated temperature or elevated CO2 or both in different soils, they induce changes in the community. So, and while observing this, I'm interested on the mechanisms that drive these changes. So what is behind the changes that we observe in the field experiments in the composition? So I hope you won't be too disappointed today, but I don't want to talk about these big experiments, but I want to focus on some of experiments that I did under control conditions to try to better understand the mechanism behind these patterns that we observe. So the first study I want to talk to is assessing the effects of ectomycorrhizal competition on both fungal and plant performance and the influence of environmental change factors on the interactions between ectomycorrhizal fungi. And this was published in FEMS Microbiology Ecology. So for these studies, we use Physolithus as our target species. And in Australia, we have 2 species, Physolithus microcarpus and Physolithus albus, that are associated to eucalyptus and acacia species. They form these earth ball fruiting bodies, and they are now distributed worldwide with eucalypt plantations. So, and this genus is interesting because it shows quite a lot of intraspecific variability. In terms of growth, you can see that they grow differently, but also in terms of nitrogen uptake, nickel tolerance, root colonization, or response to elevated CO2. So for our experiment, we used 3 Physolithus isolates, R10, Si12, and Si9. And as a host, we use Eucalyptus grandis, that it's a species that is widely distributed along the East Australian coast. And we inoculated these seedlings with either one ectomycorrhizal isolate alone, the competitor absent treatments, or with 2 isolates with, in all pairwise combinations, which are these competitor present treatments. So this was a very tiny experiment in Petri dishes where we used these eucalyptus seedlings and we placed the fungal plaques at 1.5 centimeters of the root. And we allowed the fungi to grow for 7 weeks and to colonize the plant. And we did this under 4 climate conditions, ambient conditions of temperature and CO2 at 400 ppm, and daily temperatures from 17 to 27 degrees. Then elevated temperature with an increase of 3 degrees, elevated CO2 up to 650 ppm, and both elevated CO2 and temperature to look at the interactive effects of those 2 factors. So at the end of the experiment, we measure the mycorrhizal colonization by each of these isolates when they were alone, so when the competitor was absent, versus when there was a competitor in the system. And we did this under the 4 climate treatments. And then we also analyzed, we measured the growth of each of the components of the symbiosis by itself, so the fungus and the plant. So we measured the fungal, the colony perimeter of the fungi, and the seedling mass of those seedlings, also under the different competition treatments and under the 4 climate conditions. So looking at the results here, you can see the proportion of mycorrhizas that was formed by each of the isolates here are 10 Si12 or Si9 when there was no competitor, when they were by themselves here in white, or when a competitor was present in the system in gray, and under the 4 different climate conditions. So as you can see, we found a decrease in colonization due to the presence of a competitor for all the isolates and under all the conditions. So it was pretty strong negative effects that were reciprocal. So whenever 2 fungi were interacting, both of them formed fewer mycorrhizas when there was a competitor present. And so these reciprocal effects have been rarely reported in Ectomycorrhizal fungi. Usually there is one species that is affected by the competitor and the other that performs still well. But here, all of them were negatively affected. And in particular, Si9 was almost outcompeted from the root system, while between R10 and Si12, there was no clear winner. And we believe that these strong negative effects of the presence of a competitor in colonization, so in their performance, may be due to the fact that the isolates are closely related, so probably they will have similar requirements. The competition between Ectomycorrhizal fungi also had negative effects on seedling growth. So here you see the growth of the seedlings growing with 1 isolate only or with 2 isolates. You see that there was the seedlings grew less when they were inoculated with 2 isolates together. And we ran a structural equation model with the data of this experiment, and we found that this decrease in plant growth was not due to the fact that there were fewer mycorrhizas. There was no relationship here, but it was more of a direct effect of the presence of 2 isolates on the seedling mass. And again, we think these were too similar to be providing different benefits. If we look at the influence of climate factors, looking at the seedling mass, we found that the plant growth increased mostly with elevated CO2. And if we look at the fungal partner, the colony perimeter, we found that fungal growth increased mostly with elevated temperature. So the plant was elevated CO2 had a positive effect on plant growth and elevated temperature on fungal growth. So they were responding differently to the fungal to the climate factors. We also found that there was a decrease with elevated CO2 on fungal growth under ambient temperature, but then the elevated temperature overrode this negative effect. And in terms of colonization and number of mycorrhizas, we also found that when there was no competitor, the proportion of mycorrhizas increased also with the temperature. So that it was also positively affected by the elevated temperature. Instead, when there was a competitor present, the fungi were not forming more mycorrhizas. They did not respond to the elevated temperature. And all isolates responded in a similar way. So that's why we did not observe any shifts in the competition outcomes, because all isolates were responding in a similar way to both elevated temperature and elevated CO2. And so this lack of positive response to the temperature in the presence of a competitor, this could be because the fungi are allocating resources to competition instead of colonizing the plant. And this lack of response induced a larger decrease in colonization due to the competition under elevated temperature than under ambient temperature. So after seeing this strong reduction and these strong negative effects of competition and this reduction in colonization, the following question that we ask is what drives the reduction in colonization that we are observing. So we thought about several possible explanations. So the first one could be that the fungi are competing for limiting resources. And in this case, this would be the root tips. So root tips to gain access to carbon. And however, by looking at the results, we found that the number of root tips in plants inoculated with one isolate or with two was the same, the number of total root tips, but still the total proportion of mycorrhizas when two isolates were present was lower. This is summing the mycorrhizas of the two isolates together. So in these cases with two isolates, there were a lot of uncolonized root tips that were waiting to be colonized. So it's not that there were not root tips available, but the fungi were not colonizing them. So this hypothesis is not supported by our results. So second, the fungi could be allocating, there could be changes in resource allocation in the presence of a competitor that could explain this result. So our hypothesis was that fungi could be allocating more resources to mycelium growth to try to colonize the space as quickly as possible instead of colonizing the plant. So to assess this, we calculated the investment ratio, which is the centimeters of colony divided by the number of mycorrhizas. So we found that in the presence of a competitor, there were some isolates like R10 or Si9 that in the presence of the competitor invested more in mycelium than in mycorrhizas. They were allocating more resources to grow quickly and colonize the space. But this was not the case for Si12 that has no changes in the investment ratio. So we believe that there has to be another explanation, not just this. So third, we thought about the allocation of resources to production of antifungal compounds so that the fungi would be allocating resources to try to outcompete the other fungus. And this is partly supported by the observation of deadlock. So deadlock is that in the areas of these two fungi interact, there is the colonies don't overlap. There is no fungus that gains headway. So this would suggest that there are strong interactive interactions here, and that the fungi could be spending resources in producing antagonistic metabolites such as volatile organic compounds, and then they will have fewer resources for colonizing the plant. And last possible explanation would be that there is plant control over colonization when given a choice. So the hypothesis would be that when there is only one symbiont available, then the plant would not exhibit any mechanism of plant control over colonization because there is no other choice. But when there are two symbionts available, the plant could be limiting the allocation of carbon to the fungi, limiting the colonization, and forcing them to be better symbionts, so to provide more nutrients in order to colonize the root system. And this is also supported by our the structural equation model results, because we found a direct negative effect of the presence of two isolates on the proportion of mycorrhizas. regardless of the fact that both mycelium and roots were available, and overriding any positive effect of the increase in mycelium on the proportion of mycorrhizas. So to summarize this first study, we found that the two components of the symbiosis, so the fungi and the plants, were responding differently to the modified factors. And so this could cause an imbalance of the symbiosis under future climate scenarios. Then the increased strength of negative competition effects could lead to the exclusion of individuals that may have different traits, so could potentially reduce the ecosystem resilience to future scenarios again. And we found that the biotic interactions modulate the individual response to climate factors, so they should be taken into account when trying to predict the effects on particular species. And finally, we found that ectomycorrhizal competition in our study reduced both plant growth and also colonization. And this reduction in colonization, according to our results, could be explained either by the production of antifungal compounds by the fungus, or the plant control over colonization, or both. So to explore this idea a bit deeper, we continued pursuing this idea, and we designed a couple of studies trying to test the plant versus fungal control over the results of ectomycorrhizal competition, assessing the plant nutrient exchange dynamics, and possible mechanism of plant control. And this was recently published in the ISME journal, and we also have another manuscript under review in New Phytologist. So this plant control over colonization has been described in the arbuscular mycorrhizal symbiosis. This is what is also known as partner choice. So for example, Kears et al. Show that when you pair one cooperative fungus with one less cooperative fungus, so one that provides more phosphorus in this case, and one that provides less, the plant preferentially allocates carbon to the more cooperative fungus. This is a system of reciprocal rewards that stabilizes the symbiosis, in which I provide you with more nutrients and you reward me with more carbon. And however, there are some recent studies suggesting that this partner choice would be not universal. In the case of the ectomycorrhizal symbiosis, the evidence is equivocal, and there are several studies suggesting that there would be no correlation between the nitrogen provided to the plant and the carbon received by the fungus. So for example, Kears et al. Performed this experiment in which they reduced the amount of carbon being allocated to the fungi by shading, and so they have a treatment with more carbon being allocated to the fungus and another with less carbon. And contrary to that, what would be expected, they found that when more carbon was allocated to the fungus, less nitrogen was found in the leaves. Sorry, the white is nitrogen in leaves, and gray is nitrogen in the ectomycorrhizal fungi. So actually, the fungi were retaining the nitrogen for themselves. And so there is evidence against reciprocal rewards in the ectomycorrhizal symbiosis, which then leads to the questions: what drives nutrient flux in this symbiosis, and what causes one symbiont to colonize more than another one? So one can test plant versus fungal control by growing two fungi, here in blue and in red, and in two types of microcosms. You can have a microcosm with a physical barrier in the middle, the indirect competition system, in which the fungi are colonizing the same plant, but they cannot directly interact. And then you can have the same system, but with no physical barrier, so the direct competition microcosm, in which they can also interact directly. So if you find the same colonization patterns in the two types of systems, that would be consistent with plant control. Alternatively, if you have, if the patterns of colonization change between the two types of microcosms, that would be consistent with fungal control, so that the fungal interactions are important and are actually driving the outcomes of the competitive interactions. So to test for this, we designed another experiment, again with microcosms, and we used the same plant host and the same 3 Pisolithus isolates, and this time we grew them under 3 competition treatments. So no competition, indirect competition using split plates, and direct competition with no barrier. So here you have an example of an indirect competition microcosm. So the root system of the seedling is split in the 2 halves, and there is a physical barrier in the middle, and then we grew 1 isolate at each side. And I have to say that it was pretty challenging to accomplish this. And then after 6 weeks, we counted the number of mycorrhizas, and we measured plant and fungal growth. And so to assess the nutrient exchange dynamics, we also labeled the fungi using N15. So we created a fungal compartment that only the mycelium could access, but no roots were allowed to access that compartment. This was simply by cutting the agar here and making sure the roots were not there. And so we labeled the fungus with N15, and we track the N15 that was in the leaves of the plant. And then we also labeled the plants with carbon-13, and we tracked the carbon-13 in the mycelium to look at how much nitrogen was being transferred from the fungus to the plant, and how much carbon was allocated from the plant to the fungus. And we also made sure that the fungi did not have access to carbon-13 by sealing the internal plate for the labeling. So for the labeling, we used a polycarbonate chamber in which we injected the labeled CO2 with a syringe, and that's where this experiment became a family experiment, because I got my husband here to design and build the chamber to meet our needs, so that was also pretty fun. And it worked. And so we then combined the stable isotope labeling, and also we did some RNA sequencing to try to elucidate whether the plant is rewarding the best symbionts in terms of N provision with more carbon or not. So if we first look at the results when there was no competitor, the competitor absent treatments, we found that there were differences between the isolates in terms of how good symbionts there were. So Si12 and Si9 were good symbionts. They provided more nitrogen, and instead R10 was a bad symbiont. It was not providing nitrogen to the plant. And instead, when we measured the percentage of nitrogen in the mycelium, we found that it has a larger percentage of nitrogen. So it's not that it was not uptaking nitrogen. It was, but it was keeping it for itself instead of transferring to the plant. But instead, if we look at the carbon allocation patterns, we found that the plant was making no differences. So all of the isolates received similar amounts of carbon, regardless of how good or bad symbionts they were. And in terms of root colonization, actually it was R10, the one that was forming more mycorrhizas of the 3. So we call R10 a cheater. And so the best colonizer was neither the isolate that provided the most nitrogen, nor the one that received the most carbon. And this fungus, one of its traits is that it grows, it forms a much less dense colonies than the other two, so it can quickly grow and colonize quicker, and we believe that this way it can allocate more resources to mycorrhizal formation instead of having to allocate resources to produce a dense colony. And so overall, we found no correlation between the carbon that was allocated to the fungus and the nitrogen that was provided to the plant. And then we look at the expression of nutrient transporters to see how they correlate with this stable isotope data. And so here, green represents downregulation and red upregulation of the genes. So first thing, if looking at the nitrogen transporters in the mycelium, so in the fungus, we found that R10, that was the one with the lowest nitrogen transfer to the plant, had the lowest expression of nitrogen importer genes in its mycelium. And also we found interesting that Si12 and Si9 had different types of importers activated, so that this would suggest differences in nitrogen use. They would have different preferences for different forms of nitrogen. Then if we look at what happens in the mycorrhizal tissue, so where the exchange actually takes place, looking at the fungal RNA, we found that again R10 had the lowest expression of amino acid efflux genes in these mycorrhizas. And interestingly, there were only 2 genes responsible for amino acid exportation to the plant that were activated. So despite the key role of this nitrogen transfer to the plant in the symbiosis, in our system it was only regulated by two genes. And then on the plant side, again, well, we found different sets of genes activated in Si12 or Si9, and there were many of these genes that were related to amino acid importation. So this suggests that amino acids have an important role in this transfer between the fungus and the plant. And on the carbon side, we found that Si12 and R10 got similar amounts of carbon, but they induced completely different host carbon transporter genes, which supports the idea that there would be redundancy in the role of the genes. And then we try to correlate the stable isotope labeling data with the transporters expression data using a PCOA analysis of the transporters data. And so when doing the correlations, we found a positive correlation between the nitrogen in the leaves and the overall expression of nitrogen exporters in the fungal tissue of the mycorrhizas. And then we found a positive correlation between the carbon in the mycelium and the plant carbon transporters. So this suggests that the partner that provides the benefit has greater control over it than the partner that receives that benefit. So that's why the fungus would control the nitrogen that gets to the plant, and the plant would control the carbon that gets to the fungus. Then we look at what happens when we introduce a competitor in the system, and we found some changes in nutrient exchange. So in particular, R10, the cheater, delivered more nitrogen when it was directly competing with another isolate. And we also observed an increase in this in the expression of these amino acid efflux genes that were already activated on the other isolates, but not in R10. So when there was a direct competitor, R10 was. We also observed this increase in the expression of these amino acid efflux genes. So we believe this could be a plastic response to become more cooperative in the presence of a more beneficial fungus. So if there is another fungus that is providing more, then maybe I need to also show that I can be a good symbiont. And in terms of root colonization, we found that R10 formed fewer mycorrhizas when in competition, so that the plant somehow was able to limit colonization by R10 when another fungus was present. And the fact that we found similar results under indirect versus direct competition also supports this plant control scenario. So we would be in this case here. And interestingly, when looking at the carbon allocation, we found that this was not explained by a reduction in carbon allocation. So R10 was forming fewer mycorrhizas, but still was receiving the same amount of carbon from the plant. So it was not because it was receiving less carbon that was forming fewer mycorrhizas. And yeah. And so we, while doing a stay at the Kennedy Lab in the University of Minnesota, we also designed a similar experiment to try to ask similar questions, but using another model system, so Larix and Suillus species, and we introduced soil to the system. So we also found that so two of the species were not affected by the presence of a competitor. This is no competition or another species present in the system. Two of the species were growing the same, but one of the species, Suillus spectabilis, had reduced colonization almost to 0 when another fungus was available. These, these were a split microcosm system. These were two plates glued like this. So it's a split system where the fungi cannot directly interact, only through the plant. So here, again, we see the same negative effects of a competitor, and we believe this would not be explained by reduction in carbon allocation either, because there is not even the possibility to assess if it's a good symbiont, because this is before any interaction. This Suillus spectabilis is not even allowed to colonize if there is another fungus. So then we ask how, if it's not by a reduction of colonization, how would the plant control this colonization? So we then explored the RNA data a bit deeper, and we look at the genes that were differentially regulated in roots colonized by R10 when a competitor was present, compared to when R10 was alone, when there was no competitor. So it would be the genes here in red. And we also look at, so we specifically target the genes that were not differentially regulated in roots colonized by Si9 or Si12. So specifically looking at genes differentially regulated against R10 when there was another option available. And we found 145 genes that were activated against R10, let's say, when there was another option. And interestingly, there were several sets of genes that were related, defense-related genes that were upregulated. So we believe that the upregulation of defense genes by the plant could be a potential mechanism to avoid cheaters if a more favorable symbiont is available. And this control would be localized because we found that these genes were activated in roots that were colonized by R10, but not in roots of the same plant that were colonized by Si9 or Si12. And going back to the Suillus experiment, there we also measure the time spent with Ectomycorrhizal. So actually the larger this value is, the sooner the plant was colonized by this fungus. So we found that eventually after 20 weeks, Suillus spectabilis eventually colonized the roots, even if it's at low levels. So we speculate that because these were split systems, and one of the sites was colonized by one of these other species since the beginning, then at some point nutrient depletion on that site colonized from the beginning would create an incentive for the plant to allow colonization by Suillus spectabilis, even if it was not as good symbiont as the other, to gain access to those nutrients on the other side of the system. But this is speculation. And then finally, because we are aware that these results, our study system was, there was no nitrogen limitation, and this could change depending on nitrogen in the system. We ran another experiment in collaboration with Laura Bogar and the P Lab at Stanford University, and we had three different nitrogen treatments. These are a split microcosm. Again, you see it's growing every time. With every experiment, it's a bit bigger, and it's still a split microcosm system. And so we had a treatment in which there was no nitrogen added in any of the two sites. There was another one with addition of nitrogen in form of casein in the two sides of the system, and addition of nitrogen only in one of the sides of the microcosm, to force these differences in nitrogen availability in the two sides of the system. And then we look at carbon allocation to these two sides. And what we found is that in what we call the choice treatment, so there is nitrogen only in one side, the plant was allocating more carbon to the roots that were on the side with access to nitrogen. And it was doing this with no distinction between roots or mycorrhizas. So was allocating more carbon to roots, colonized or not, that contain more carbon. So the larger the difference in percentage of nitrogen in the roots of the two sides, the larger the difference between the two sides, the larger also the difference in carbon allocated to each side. But this was independently of whether the roots were colonized or not. The plant was just allocating to the site with access to nitrogen. So, to summarize, we found that when given a choice, the plant had the ability to limit root colonization by the least cooperative symbiont. This was not explained by a reduction in carbon allocation to the fungus providing fewer nutrients. Instead, we think that partner choice in the ectomycorrhizal symbiosis could operate through the upregulation of defense compounds that would prevent infection and discourage cheaters from the system. We found that ectomycorrhizal competition might encourage mutualistic behavior in some cases. And finally, that under some circumstances, plant can direct carbon towards portions of the root system that provide greater nitrogen. So overall, the fact that the plant can discriminate among partners explains allows the host control over ectomycorrhizal colonization, and explains the key role that we think plants have in structuring the ectomycorrhizal community. And last, I would just like to thank my co-authors and team at the Hawkesbury Institute of the Environment, funders, and all colleagues also at Minnesota and Stanford, others, and students and technical staff to make these experiments possible. And thanks to all for your attention and being here.
Questions? Any questions? Yes, Jordan?
Yeah.
Is it possible in this second and third experiment that the isolates that you call cheaters actually interact with the plant through another nutrient? So it's not through nitrogen?
Yeah, this is what I didn't mention, that actually, so we always say this is bad symbionts in terms of end provision, but of course ectomycorrhizal fungi provide other benefits that we did not measure. So this, I forgot to say, thanks for the question, that actually the plant did not completely repudiate R10, or the cheater. It still allowed some colonization. So this could be either because the plant cannot totally control it, or because this fungus could be providing other benefits that we didn't measure, of course. So, yeah.
Do you know how common is it? Could it be that it's important in terms of phosphorus, for example? It's unlikely because the mechanisms are similar.
Yeah, so the main nutrient in ectomycorrhizal symbiosis is nitrogen, but there is also evidence for phosphorus being important. And we actually, on that experiment, we did first, so in the first, the study number 2, the one with our R10 and Si12 and Si9, we also tried to label the fungus with phosphorus, radioactive phosphorus. Well, we did it, and we did not find any differences in provision of phosphorus to the plant. It was actually the detection of phosphorus in the leaves was quite random. We just found it in some cases, so it was not enough to make any conclusions. That's why we did not finally include the data. But we tried because we believe that phosphorus can be important also in some conditions, and of course other minor nutrients that we don't even think about them. But so we tried nitrogen and phosphorus, but phosphorus, it was just randomly in some plants, but we did not detect any pattern of why it would be important in ones or not. Yeah.
Actually, I have another question.
Yeah, go for it.
On the first experiment, I think you said that the response of the different isolates there was similar to elevated CO2 and temperature, but you did not find evidence of a change in the composition.
Yeah.
But can we see again the...
Sure.
Because it seemed to me that at least in some cases there was a big difference.
Yeah, and actually one of the isolates continued to respond negatively to elevated CO2, but let me
the
see.
first and second were changing ranks, I think, in some cases at least.
So
Here, but there was...
this is fungal growth. You mean on the micro? So here we see that elevated CO2 was negative, had a negative effect under ambient temperature for isolates, and with elevated temperature there was an increase in all of them. But still there was a negative effect of elevated CO2 only for Si9. The other two were similar, so temperature overrode the negative effect of elevated CO2 in two of the isolates, but Si9 still continued to respond negatively to the elevated CO2. And then
here, for example,
here.
you have between R10 and Si12. Si12 seems to perform better at ambient conditions, but then R10 performs best when the competitor is absent under elevated CO2 and ambient temperature. No?
Elevated CO2 and ambient temperature.
Yeah, so the second column.
Yeah. Yeah, the thing is that there was a lot of variability in this
data.
data.
R10 more or less is the same.
Yes.
It doesn't seem significant.
Yes.
Si12 clearly seems to go down. No? When you
Yeah, by the, yeah. Yeah, there was actually a negative effect of the CO2. Let me just check. Yeah, it was not significant, but yeah, because there was quite variability in the data, but yeah. I see what you mean. Yeah.
I have a question.
No. Go ahead.
I have a question. Whether this kind of the plants inject this fungi, whether have the temporal scales, for example, 1 month, 2 months, or 3 months, whether they have some difference when you have this measurement.
Sorry, I didn't get that. So can you repeat
Okay.
it? Yeah.
Just the plants, if you inject the fungi, for me, I'm very curious about how about the temporal scale changes. For example, you see the growth, the computation, maybe for the first 1 month, the computation not very strong because the resources are very rich, but after some months, maybe the changes and
Yeah.
the computation very strong, and then, so maybe the response are different. So whether you consider about this temporal scale changes?
Yeah, in this first experiment, it was really, we couldn't do, it was just a 7-week experiment. So really it was already a very short-term experiment. The problem is that these are quite short-term experiments. You see in the other a bit longer experiments with Suillus, what we were saying here that so actually this was we were looking at the mycorrhizals at different weeks, so we track what was happening every week, and as you say, at some point this might shift because this fungus could start colonizing the system. So we don't know after, if it was a longer experiment, if this could shift, and actually that fungus could colonize more than the others, or so. Yeah, I guess interactions outcomes may change with time, but these were pretty short-term experiments, so it's difficult to see these patterns. It has also been shown that it's different if you inoculate the fungi as mycelium, so that they can already start colonizing the plant, or as spores. And this system was we inoculated the fungus in form of a spore. So then it's also different because they have to first grow and find the host and colonize. So it's also different how you inoculate the fungi. Yeah.
Just as a curiosity, in the experiment where you have the plate divided in two, you say it was a little bit challenging.
Yeah.
To the roots to the.
Yeah.
It was really difficult to shift the roots in these two parts of the plate.
It's easier with conifers. So yeah, here. So first we grew the seedlings from the seeds. We allowed them to grow a little bit, and then we cut the main roots to induce the formation of lateral roots that could be split in two. In some roots did it, so we have to discard some seedlings. And then, well, we had to make a hole here and put half of the system at each side, like similar proportions. So we finally designed this system, and we had a larger plate that included the host system. But of course, we had problems with contamination, as you know, I'm sure. And so because this first plate was not completely sealed, only a little bit here, and the external plate was sealed with parafilm. So yeah, it was quite a lot of manipulation, so that could induce contamination. So we had to grill them for a while. So we were always looking at them. Please, no contaminants. Wait a little bit because we have to do the labeling. So, but yeah, it was fun. Yeah.
Do you think plant genotype could influence the results as different strains of fungi?
Sure. Yeah. It's too many variables to. Yeah. For this experiment we used. So we bought the seeds that were kind of controlled so that they pretty much behave the same, but yeah, depending on also from these are species that grow all along the Australian east coast, and depending on, there is a lot of variability within eucalyptus that it's been shown. So of course, if there are differences between individuals of fungi, I'm sure also the plant genotype, it's also a thing that it's being studied a lot in the Hawkesbury Institute. There is some people looking at this variability depending on the origins of the species. So yeah, so but we try to control a little bit for that.
I missed the part in which you explained how you managed to label the mycorrhiza but not the roots.
Yeah, here. So we actually cut the agar here. So we removed the agar, creating a kind of a bridge that the fungi, because they grow kind of diffuse, they could still grow through the bridge and reach the other side with the agar and then continue to grow in the agar. And so we place here a paper filter with the label N15 solution. And before doing that, we made sure that there were no roots accessing this, what we call the fungal compartment. So if there were any roots, usually they were not, they were staying in this part here, but if there were any, we just trimmed them to make sure that any nitrogen that we got here came from the fungus and not from roots that were accessing this label solution. That was another step that could introduce contamination, but that we did to make sure that there were no roots accessing that part. And for the carbon labeling, before doing it, we seal all this internal plate with electrician's tape and with plasticine to make sure that we didn't break the stem. So when we made sure that all was sealed so that the air was not going inside the plate. So that any carbon that we detected here came from the plant. And we had all the controls to make sure that there was no nitrogen diffusion here or carbon diffusion to the fungi. Yep.
I have a question. Your main findings is that plant control.
Yeah.
Interesting. But my question is, how is that control? Is before the infection,
Yeah.
the infection, or after, or it can be at any point?
Yeah.
The literature says.
We believe it can be both prior to the infection, and it has already been shown that ectomycorrhizal fungi and plants exchange molecules before any interaction takes place, like these small peptide proteins. So we believe this control can be prior to the symbiosis, because that would explain why this fungus is not allowed to colonize when there is another one, and this has not even, there is no no colonization. And these last results here, when we see that the plant can allocate more carbon to one side, makes us think that maybe there is also some control later once the symbiosis has been established. So, yeah, we believe it's at both stages with different regulation systems. And this is so our results support plant control in these cases, but this does not rule out the option that there is also some kind of fungal control in some cases with these antifungal compounds. We did not find any overexpression of antifungal genes, compound genes in our data, so we did not find evidence for these metabolites being important, but of course in other systems or conditions they might be, so yeah.
So thank you very much, Sara.